decoding our blueprint. So the idea of completely reading out the letters of the human genome, which basically meant decoding our blueprint, came to sort of crystallization in the late 1980s. And the reason for that was the recognition that if we could get foundational information about our blueprint, it would set up a series of studies that would help us better understand human biology and eventually human diseases that might lead to our ability to improve human health. And I often make the point that some of the earliest implementation of genomics in the medical situation is going to be with cancer. Cancer is fundamentally a disease of the genome. I mean, the reason a tumor grows is because the cells in that tumor have picked up glitches, they've picked up mutations, they've picked up changes in the DNA that make those cells grow out of control. It's like pressing an accelerator in a car and just keep it going. It just grows and grows and grows and grows. And the reason why is because something's broken in the genome. I think standard of care for many types of cancer are going to be get that tumor, read out its DNA, sequence its genome, and based on what you've seen, what's wrong with that tumor, not by looking at it under a microscope, but actually looking inside its blueprint, you will be able to have a much better way of deciding what types of treatments to pursue and have a much better idea about what's wrong. But that was an audacious task. It would require an industrialization, if you will, a scale up of a scientific effort that was really unusual for biomedical research. You know, biomedical researchers traditionally, they have laboratories of 6 or 10 people. They work on profoundly difficult problems, and they like to explore. They like to sort of ask big questions. This is very different. This was a very focused effort, very defined agenda. Sequence the human genome. To do that was going to require multiple countries. It was going to require thousands of scientists, and it was going to require a number of years, originally slated for 15 years and ended up taking 13 years. That is why the analogy to putting a man on the moon has often come in, because the idea that was a very focused goal. We are going to do something that's going to be a grandly large project requiring many people with a very defined goal. We're going to put a person on the moon. And so that was something that biomedical researchers looked at and said, wow, we never do anything like that until the Human Genome Project.